coming. Today we have a very generous offer from Stoeckle to talk to us about DNA barcoding. So we're actually for people who are just popping in, we have a 5-day course happening right now on DNA barcoding that is being run by Cold Spring Harbor Labs. So we have several teachers with us who are learning DNA barcoding so that they can use this technology and bring it into the classroom and try to engage kids with these types of projects. So Mark, who has a lot of experience with DNA barcoding, is going to be discussing with us his understanding of the research and also some exciting projects that he's been involved in, involving cockroaches for one. So thank you, Mark, for your time.
Thank you, Gene. Can we have the lights down a little bit?
Yeah, we could do it from here.
Oh, sorry.
Sorry about that. Okay, whoops. That should be good. How's that? Is that okay?
Okay, great. That's great. Thank you. I'm going to talk a little bit about DNA barcoding, so simple way of identifying species by DNA, and a little bit about what, why, and where, how it started, some very fun student projects that we've been involved in, and some recent work we've been looking at large-scale evolutionary patterns using this. So, definition, what is a DNA barcode? Just a short, standardized gene region for identifying animal, plant, and fungal species. So by analogy with the UPC code that you see on product labels that can be scanned by a machine very quickly, the idea is read a certain segment of DNA from, same segment of DNA from different organisms and scan that and then be able to match that to a species. So why do we need this? First of all, lots of things that we would like to identify are in bits and pieces. This is a photomicrograph of bat guano. There are lots of little insect parts in there. If you want to understand food webs, you want to understand what animals are eating, you often want to look at the remains that they leave behind. If you're really good, you can tease out bits of insects from there, and you might be able to, there's an insect wing there, you might be able to assign it to one group of insects or another, but there's enough DNA in those that you can identify actual species of insects, and you can figure out what the bats really are eating. Second, lots of things are immature forms that we'd like to identify. These are fish larvae, these are zebrafish larvae, but they look a lot like all other kinds of fish larvae. So when things are eggs or when they're seeds, that's often when we'd like to identify them, and you just can't do it by morphology. And third, there are just a lot of species. This is a picture from a lighted sheet that was put up in a forest in Costa Rica, but you could get a similar result in the United States. These are mostly moths. There are 160,000 species of moths in the world. And the best an expert can do is to be able to distinguish a few thousand species. So there are just too many things out there for anyone to identify them. Now you can sort them out and then deliver them to the various specialists, but that takes a long time. So we'd like to get DNA to help us here and computers to help us. Where can we apply it? I'll talk about 3 areas: human health, for the environment, and for scientific discovery. So human health, this is a hamburger. You might want to know what you're eating when you eat a hamburger. You can't, it says on the label what it is, you don't actually know. And one of the reasons you might want to know, you remember earlier this year, it turned out there was actually a lot of horse meat in products that were sold as ground beef. And the way that we know that is through DNA testing. So even when the food has been processed like this, ground up, and even after it's been cooked, there's enough DNA there to get a short segment of DNA barcode out of that and figure out what it is you're actually eating. For disease vectors, this has a lot of applications. These are 2 species of mosquitoes that live in New York City. One of them is a vector for West Nile virus, the encephalitis virus, and one of them is not. This is Culex pipiens is a West Nile virus vector, and this one's actually a different genus, is not. Now, a mosquito expert can tell those apart, but there aren't that many mosquito experts. So, and they can only go sort out specimens a little bit at a time. But you can imagine you can automate this. You can have a trap. It can collect DNA. It can collect lots of samples. You could actually analyze them as a pooled DNA extract and figure out whether you have the vectors there. And not only do you want to identify them when they're big, you'd like to really identify them when they're little. This is an egg raft of this same species. It's tiny. So no one can tell what that is. You can say it's a mosquito egg raft, but we don't know what species it is. And this is the stage, if you're going to do spraying, that you'd like to spray. And if it's not the target species, then you don't need to spray. There's a lot of trade in endangered species. These are products that were seized either in New York City last year, in China at the Russia-China border earlier this year, and in Brazil last year. These are shark fins, these are bear paws, and these are parts of various primates. Now, some of these things, so there's a lot of this going around the world. Many of these are endangered or protected species. Sometimes you can identify them. They're intact enough that you could tell what this is from. Shark fins, generally, you can't tell one species from another. Some are protected, some are not. So there's been a big effort, and there's actually a dedicated project, the Barcode of Wildlife Project. And the idea is to make sure that in the library, we have barcodes for all of those endangered species and all of the things that they're closely related to. So if you're doing a prosecution, that you can go to the database, find a reference sample that's been authoritatively identified, know that it's not a closely related species. And this effort is being run by the Consortium for the Barcode of Life. It's based at the Smithsonian and received just recently an award from Google, a $3 million award to support this project. So the whole thing started really in 2003. Paul Hebert, who was pointed to with the arrow there, Canadian scientist said, wouldn't it be nice if we could use short DNA sequences, which he called in this first paper DNA barcodes. Wouldn't it be nice if we could make a database of those and identify species? And the original paper was proposing the idea and some sort of tests, just looking at what was in the databases as to whether it was feasible or not. This led to 2 conferences at Cold Spring Harbor, where said, okay, it's a good idea, but can you make it happen? Because it involves getting a lot of people to work together and to agree on standards. And after the second conference, it was decided, yes, it's worthwhile, benefits to society, benefits to science, and we should go ahead. So the idea is really very simple. It's just you get some tissue sample, you put it in your barcode analyzer, a DNA sequencer, you get out your sequences, and then you look in your library and see what does it match to. Now, obviously for this to work, your sequence has to be in the library. So if this is a database of criminals, if you find a fingerprint doesn't match to anything, then you don't know anything. So a big part of the effort has been to build up this library of reference sequences. So what's an ideal DNA barcode? It would be present in all organisms. We don't want to have to identify the organism first to decide what gene we should barcode. So it should be present in everything. Ideally, it would distinguish all species, and it should be easy to amplify and sequence. So that you can sort of stick your tissue sample in the machine and get back a DNA sequence. We're fairly close to that. We have 3 sets of standards for animals, part of the CO1 gene, mitochondrial gene for plants. It's a combination of 2 genes, chloroplast genes, MATK and RBCL, and for fungi, a nuclear gene, ITS. So I'm going to talk mainly about applications for animals. So CO1, this is mitochondrial genome. It's about a 15th of the whole thing. It's a short, about half of the CO1 cytochrome oxidase 1 gene that's present as a separate piece of DNA inside the mitochondria. And it works really well, distinguishes about 95% of species. So the other 5%, you might say, well, it's one of these two or it's one of these three. I know it isn't anything else besides those, but it won't tell those two or three apart. So the big challenge that I mentioned is building up the library. And there are just a lot of species out there. For things we've already named, 1.9 million, about 1 million of these are insects, about 70,000 are vertebrates. So this is an ongoing effort to build up the library. And obviously someone has to get a specimen, identify it, so it has to be by an expert. So using the names that we have, store it in a museum so you can go back and check your results if they don't make sense, and put the DNA sequence in the public databases. Okay, so that's the background. I'm going to talk about 3 student projects that I've been involved in that have been a lot of fun. So these are, I'd say these are all voyages of discovery. You can do science by making a hypothesis and say, okay, and then testing that hypothesis, or you can get on a ship and go somewhere or go to the bottom of the ocean and and see what you find. So these are more in the voyages of discovery category, but in each case the students found things that no one knew were true or no one knew existed. So first, my daughter, Kate Stoeckle, and her friend Louisa Strauss, when they were in 11th grade at Trinity School here in New York City, said, I was working on the DNA barcoding project, and Kate and Louisa liked to go eat sushi after school. And they said, well, can we identify it with DNA barcoding? And I said, I think so. And I said, I don't think anyone's done that. It's not like a scientific field, sushi. So I said, that sounds like fun. Why don't you do that? So what did they do? First, they went shopping, and they bought different kinds of sushi and fish products from stores in our neighborhood. I helped them take little samples. There's a little bit of fish from one of the pieces of sushi, put them in a mailing kit, make sure that we document everything so we know what samples are which, and then send them off for sequencing, and then get the results back by email. And then, one by one, we took those sequences. I think you've been doing this, and matched them to the database. We're using there's a BOLD, Barcode of Life Data Systems database, or GenBank. They're overlapping databases. So what did they find? They found a quarter of the samples that they purchased were mislabeled. And it was always as more expensive or more desirable fish. It seemed to be pretty common. It was found in 6 of 10 grocery stores or fish markets and 2 of 3 restaurants. So, for example, something that was sold as white tuna, it should be albacore tuna, which is $8.50 wholesale at that time, turned out by DNA to be Mozambique tilapia, freshwater fish, very inexpensive freshwater fish that's farmed. And if you look at, you can probably tell tuna and you might be able to tell salmon, but most other kinds of fish you can't tell apart. So this got a lot of attention. Actually, Kate and Louisa wrote a short report that was published in a trade journal. You may not have seen this, but it's Pacific Fishing. It's a trade journal for fishermen. They published Kate and Louisa's report. And from there, got a lot of interest. They were on page 1 of the front page of the New York Times. They're on morning television. This is my daughter Kate and myself discussing the sushi project. And the story really went around the world. It was in newspapers and magazines and websites around the world. This is a cartoon from the Korean Daily News, has a circulation of 1 million. Shows 2 girls looking at a fish on a plate with a magnifying glass and the chef being led away in handcuffs. So that's kind of the story of the project. And in Time magazine, this is their quote, "If you're paying for white tuna and you're eating tilapia, I think you'd want to know that." There's continuing interest in their project. This last year and this year, their story appeared in 2 college biology textbooks, one by McGraw-Hill. They gave them 2 pages, put their pictures in it. There are not many scientists in this textbook. But there's their fish story, telling a story of their investigation and what they found. And this year from Pearson, similarly, and there's their picture and about their DNA investigation and about DNA barcoding. So a lot of interest in the story. I'm better known for being associated with my daughter's project than anything else that I've ever done, and that's going to continue. It's just an easy story for people to understand what DNA testing is about and what you can learn from it, and the idea that it's pretty simple, at least the DNA barcoding part here. So that project was a lot of fun. The next year we went back to Trinity and I said, or looked for student volunteers and said, let's just, why don't you test anything you can find in your house or the neighborhood that might have DNA in it. They got things like a feather duster, and we got ostrich DNA out of the feather duster. They did different kinds of food. They did insects that were lying around indoors. And they were, this story is also reported in the New York Times. They were interviewed on NPR Science Friday. This is Brenda Tan and Matt Cost. Part of their findings were finding mislabeled food. And this was their high school students are at the lead paragraphs of an article in the Washington Post about food fraud. The idea is if high school students can find it, it must be pretty common, and we probably should do something about it. They found, it turns out you can get from cheese, you can get the DNA, there's enough DNA in there, DNA barcodes in there to tell what animal it came from. So something labeled as sheep's cheese was actually just cow. Something labeled as sturgeon caviar turned out to be Mississippi paddlefish. And one of the things they did was they found a cockroach that is genetically different than what was in the databases, enough so that it might be a new species, and that's New York Post. That was their idea of the most important part of their findings, and we'll come back to that. So the next year, we went back and students, the idea was to do something with plants. So this is Grace Young and Kate Gamble and Rohan Kirpikar, who were juniors, and Grace was a sophomore at the time. Went around and just bought tea from different stores, Starbucks, grocery stores, school cafeteria, commercial products of teas, and with the help of New York Botanical Garden, did DNA barcoding on this. So the most striking thing was that a third of herbal teas had unlisted ingredients. So sometimes it was chamomile, which is commonly used in teas. It just didn't say that on the label. But some of it was lawn grass or various other common weeds you might find in your backyard. Nothing poisonous, but those little bits of sticks and things, you really don't know what they are when you're buying tea. This was published, and the students are co-authors in a peer-reviewed report in Nature Journal Scientific Reports. And I just checked last week, this article is in the 98th percentile for what they call online attention among 10,000 articles that were published around the same time in other journals around the same time. So a lot of interest in a student project. One of the things we did for most samples were done at New York Botanical Garden. We did some of this in my dining room and kitchen. We got some, this is do-it-yourself DNA work. You will have been, I guess you've been doing this the last few days, but we got a used thermal cycler, gel electrophoresis, bought a new QIAGEN kit, and did the DNA isolation at home. It's not quite a toaster yet, but I think DNA analysis is going in that direction. Then we mailed these off to Macrogen for sequencing. So it's a very robust technology, a lot of interesting things you can learn. Those student projects were one of the things that inspired the Cold Spring Harbor's Urban Barcode Project, 2011, 2012. This is public and private high schools in New York City, 75 teams, 218 students, 30 high schools. The grand prize winners got $10,000. They looked at ginkgo biloba products, which turned out mostly to be rice. No, you may have heard this story, no ginkgo biloba. And this year, again, a good turnout, a wonderful array of projects done by students. The winner was, they looked at ant biodiversity in a park across from their school. And among other things, they found 2 species of ants that had never been reported in New York City. So they found something new. Okay. Those are student projects. I'm going to talk just a little bit about some work with large-scale patterns. Now, we have a lot of DNA sequences. What did we learn from those? And this is work done with Cameron Coffran here at Rockefeller and myself. So the part of the genesis for this is when you get a lot of DNA sequences, we usually use them to make a tree to show how things are related. That's good, but when you get a lot of organisms or a lot of terminals in your tree, you know, it's hard to see, is there any structure there? Is there anything in that? Or is it just, we can follow lines and see, but we'd kind of like to know how the relatedness of things in that tree. So this is a different way of looking at DNA sequences, which we call a Clay diagram after the artist Paul Clay. You may know he made paintings with different size, very geometric paintings, some with a lot of different colored squares. This is a matrix of comparing 400 nucleotide sequences, and we're comparing them to themselves. It's a little bit like one of those old things where you wanted to find the distance between Boston and New York. You read across and down, and you can find out how far apart things are. In this way of representing it, if it's red, it means they're very similar or identical. And we group the sequences here according to similarity, and then we look at how many groups do they fall into. So we have 400 sequences. This is a kind of skipper butterfly that's thought to, one species name, but it's thought to actually represent 10 species. And when we put them onto this graph, we actually see very distinct blocks. These are about 50 sequences that are almost identical, like an island surrounded by blue water, because they're different than all the other 10 species here. These 3 are quite closely related, and actually some people have said these really should be considered one species. So now we have like a map, shows species or islands similar among themselves, distant from what's around them. We can look at same kind of thing and apply this to warblers. These are like yellow warbler, chestnut-sided warbler. These are the North American birds that migrate through Central Park in the spring and the fall. there are 24 species in this data set. And again, you see the same impression of red or brown islands surrounded by a lot of blue. So species are distinct units. This is a graphic way of representing that. And you might say, what does it show you? Well, species are species, you know. So what we talk about, at least from this genetic point of view, whether you're talking about butterflies or you're talking about birds, they're the same kind of entity. Yes?
How do you order them on the
Excuse me?
How are the sequences ordered on the axis?
They're ordered by neighbor joining. So they're grouped by similarity. If you don't...
so that you designate one as reference sequence and then...
No, you just make a neighbor joining tree and then have the rearrange your sequences to follow the whatever order is in the neighbor joining tree.
But and how do you determine the... I mean, the neighbor joining tree is also arbitrary, right?
It is. So this has some arbitrary, you know, you can flip terminals. So the fact that 7 is next to 8 is not important, but the fact that 7, all of the things that look like 7 are together. And this set is really, these are 4 species of warblers that are quite closely related. That's looking at how differences within species are small compared to differences between species. And we can apply that same thing. We can go up a level in evolutionary analysis, and now we're looking at one individual of each species. This is 704 bird species. There are 10,000 species in the world, so there's about 7% of the known birds. And we're looking at same method, 2 different genes. This is a nuclear gene, and here's our barcode gene. And we just look at the nuclear gene first. We see that birds fall into, according to this, there are 3 islands here. There are 3 main groups of birds. And if we go back and look and say, what are these? Well, these turn out to be things that taxonomists have named. So this island of relatively closely related species are the Aucine songbirds. This is robins and wrens and warblers are here. Their closest relatives are the New World flycatchers. And then this is all the rest of birds. So owls and gulls and loons, sandpipers. So this says, well, really birds they're not just in 200 families, but those families fall into these very discrete blocks. And we can see the same structure here in this barcode region, which is, it's just much fainter. But we get the same kind of picture. So what does that mean? Well, it suggests that an idea that evolution proceeds by bursts, or what people, Stephen Jay Gould said, punctuated equilibrium. I think that's partly what we're seeing here, that this was, once the flycatchers developed, they diversified into a lot of species. And sort of not too much happened until the first songbirds, which were probably crows, were in that lineage. And then that was a very successful idea, and they diversified into a lot of closely related species. These are all the older birds, and they're not as closely related to each other as these are. And none of them are as successful in terms of number of species. So we think this is an interesting way to look at barcodes and other sequences. Okay, I'm just going to finish what we're doing now. This is from the student project I mentioned. It looked like we had a genetically distinct kind of cockroach, and specifically an American cockroach. This is the really big one, like a water bug. Some people say it's an inch or an inch and a half long. Usually lives in basements. Most people really hate seeing them. But these, we got some from my apartment building. on the west side, and they were surprisingly different, about 4% sequence different from what was in the databases for this cockroach. And the other thing we noticed at that time was there really wasn't much data on this cockroach. It's one of the very widespread, meaning around the world, pest species, and there are less than 24 sequences, barcode sequences for this cockroach in databases. No one's looked at them. They're sort of icky, they're pests, they live in urban areas. People tend to want to go out and study things that are out in the rainforest. But it seemed like a good opportunity for a student or citizen science project. So the idea was to encourage people, and I hope we have some contributors here in the room, but if anyone hasn't contributed and they see one of these, you're welcome to step on it and mail it to us. We don't want live ones, actually. So I had a friend who I said, could you get, and they said, oh, we've got one. And I left it with the doorman, and I went to the building, and they hadn't quite sealed the envelope, and it was gone. So you got to step on them. So the idea was to get people, rather than us going around and collecting everything, to get people to mail these in. And why we want to do this, well, we're saying, are there different genetic types? Was this just a fluke, this initial observation? And then can we, if there are, do they differ between buildings at a very small scale, or do they differ between cities? And maybe a big question is, are these, if there are different genetic types, do these actually represent separate species? So this work is being done. First of all, I have to thank all of the people who've collected or mailed in cockroaches to us. Joyce Zias in the Rockefeller Summer Program is doing the laboratory work in Daniel Kronauer's lab with Christoph von Buren, a postdoc. So they've, and they're taking very nice pictures of the cockroaches and doing the DNA barcoding of these. So here's some specimens. These are some sent by high school students from Spain. I don't know how they found out about it, but we did put this out on the internet. And they found some in a kitchen. And this is a restaurant. This is the north coast of Spain. But in any condition is good. This is one I found actually this morning on the sidewalk outside of Memorial Hospital. So you can't look around when you're picking up a cockroach, but you don't understand what you're doing. But I always carry a plastic bag so I can pick them up. So that's how, and so far we have about 100 specimens, including from Australia and Spain. I showed you mostly the east coast of the US. New York, here we are on the east side of Manhattan, Upper West Side, some downtown, and also Bronx and Brooklyn. But we're very anxious to have more sites. So we'd like, and there's no real closing date for the project, so we'd like you to mail them in. They're tropical species. They're thought to have come from Africa originally, but no one really knows. So you'd say, well, how do they live here? They live here because they're in cities, there are things like steam tunnels where it's like the tropics. So we've created an environment where they can live year-round. And in the summer, then they come up upstairs, and that's when we see them. And here's a little closer view. Here's Rockefeller. We got them from different buildings at Rockefeller, and here's where we're having the lecture right now. So we got one, not on this level, but the level up. So we're looking and seeing, did the Rockefeller building, any differences there? There's Memorial Hospital. So far, it does look like there really are different genetic types. So this is a graph. The colored bars, each line is an individual cockroach that we've done DNA barcoding on. The colored lines show where that individual differs from the others in this data set. And this is a neighbor joining tree over here. I think you can see there's one set of individuals that are pretty similar, up here. There's another set here that are pretty similar, and then there's a third set down here. So far, everything falls into one of these groups pretty closely without a lot of variation within the group. And this includes, so we have 35 sequences from the project so far. We have at least 100 specimens to go, and this includes what's in GenBank. So I think GenBank sequences include just 4 from the United States. That's all that's in the databases. The rest are from, mainly from Iran, Tehran. Primarily someone there wanted to study variation in cockroaches. But I can tell you there are cockroaches at Rockefeller that have a barcode 100% identical. It's one of these types to ones that are walking around in Tehran. So that's interesting. How did they get there, and why are there 3 types? So that's it. I hope this is fun, and I hope you have fun with your own projects with this. I think there's just really a lot of possibility of things you can do that are, and discover something that no one knows, either by setting up a hypothesis or just getting on a ship and seeing what you find. Thank you. Yes, Jane?
How different does it have to be in terms of its genetic sequence to become a new species? You know, is there movement towards subspecies, or where is that?
That's a really good question. It's probably the #1 question people ask. It doesn't have a simple answer. Usually, I can tell you in birds, which are probably the best studied group, if birds differ by more than 1% in their sequence, then either they are different species or they're about to be named as different species. Some things are still being split. For insects, I don't know enough. To say that these are different species, I think we'd have to show that they don't interbreed, for one. So one of the things being done in Daniel Cronauer's lab is looking at not just the barcode gene, but other genes. So you need some other biological evidence to say that the genetic difference you observe is associated with behavior that is what we call a species. A rule of thumb is 2%. It doesn't, not everything with more than 2% different is a different species, but in animals, it's usually true. Okay.
Is anyone doing anything with microorganisms?
That's a good question. The barcoding itself is not aimed at that, sorting them out. There is CO1 is, when you say microorganisms, if you mean prokaryotes?
Bacteria as well as unicellular eukaryotes.
Certainly people are doing algae and prokaryotes, I mean not protists, so single-celled organisms, and CO1 works pretty well for those. There's incredible diversity there, as you probably know. Yes?
Yeah, and just building on that, I think that there are these initiatives to sequence the human microbiome, right? So it seems like it would be an interesting thing to swap the belly button and then figure out what bacteria are living in there. And if you can apply that, and how would you do that? That would be really good projects for a lot of people.
There's definitely a lot of room there. Bacteria do a lot more sharing of genes than warblers do, let's say, or even cockroaches do. So when we call something E. coli, it actually, that's like a group of things. They're very similar to what we call Shigella. And they share genes back and forth. So if you analyze any one gene, you might not really end up putting it in the same place. It's harder to make a bright line and call something a species in bacteria. That doesn't mean you shouldn't analyze it. You'll find something interesting.
Part of the reason I ask is because a lot of what's going on right now is this environmental
Yes.
I think most of us probably have more than 1% differences, even though we're using the exact same thing. So how do you deal with when you go to GenBank? I'm sure people have made mistakes and the sequencing isn't perfect. When you then said, 1% or 2% is kind of the margin of error.
So you're saying between your forward and reverse sequences, well, first of all, cut off the ends of the sequence, because that's usually where the trouble is. I mean, it's a technology. No technology is perfect. And there is no simple way to distinguish errors, sequencing errors, from biological variation. If you repeated it and you got the same, you'd have to get more data. You'd either have to repeat it. If it turns out it puts in a stop codon, then it's probably an error. Or if it's a gap in the sequence, if it's not a pseudogene. That's a good question.
Sorry,
Yes.
can I ask another question?
Yeah.
So, like in the case of the teas, you found sticks and weeds and grass and so forth, but you don't know what percentage of, or do you have any way of knowing that it might be a very small amount, it might be what's considered to be an acceptable amount of trace, like in every food, there's
Yes.
poisons and God knows what,
Yes.
rat feces, whatever it is. And that's considered acceptable by the FDA as long as it's less than 1% of the total or whatever the number is. So, but is there any information about abundance, or is that not something you can even?
We didn't try to look at that. That's a key question. It's not simple to do by any technology or genetic technology to sort out mixtures. It's certainly a thing, and we've talking about environmental sampling, it's an issue there. I mean, the way it was done was to pick out bits out of the, you open up the tea bag, put it on the table, pick out bits.
Stuff that looks different.
And if it looked different, I mean, it's not 1 in 1,000, I don't think, but we don't know. And didn't find anything poisonous. So, yeah.
With the ginkgo teas that he was talking about, like they barcoded mostly, it was rice, but it was like the majority of the sample because when you sequenced it, that was the PCR product that was the most abundant. So it would, in some of these experiments, usually the most abundant of a mixture would be the only one that primarily gets sequenced. And so it could tell
Right, but
you
it's
the
not, it may
majority.
be most abundant, it's not quantitative.
Right, no,
Yeah.
PCR.
Yeah.
yeah, no.
Yeah.
But in any case, I mean, if it's mostly rice, it's mostly rice. It's not what it's advertised to be. So that's
Right.
the bottom line.
Yeah.
But in a case where there's weeds or grass or something, it can seem like it's a big deal to somebody who doesn't understand it, but it may not be. The abundance may be an issue,
Yes.
or may be less abundant than the actual main ingredient.
Yes. Yeah. Yes.
I wonder if your daughter and her friend became persona non grata at your local sushi establishment.
They're not that recognizable. They do get still get Facebook inquiries. There was a sort of an indignant response in the Times the next day from the chef at Le Bernardin, who said, "This is impossible. We would never make this mistake." And then there was a response to that by someone saying, "Experts are the easiest people to fool," which is true if you do wine tasting or anything. Oceana issued a report last year looking at fish labeling in cities across the U.S. And this is, they're early, and one of the things that they cited was Kate and Louisa's study, but it's absolutely, this is it's between 1/4 and 1/3 of fish are mislabeled. Oceana also did something where they showed chefs fish and, did that method of doing where they knew, because they'd filleted the fish, what it was, and they can't tell. If you get a white, you can tell swordfish, you can tell salmon, you probably can't distinguish species of tuna. You can't really tell most of the white fishes apart. Okay, all right, well, thank you very much.